The topic of this video is the replication fork of prokaryotic DNA. The process of replication can be grouped into 3 steps: initiation, elongation, and termination. This is an image of the circular prokaryotic DNA, but what exactly are we looking at? So let's zoom in. Here you see a double helix. The strands of the double helix run antiparallel in polarity, with one strand running 5' to 3' and the other 3' to 5'. The nucleotides are held together with phosphodiester bonding between the 3' OH group of one sugar molecule and the 5' phosphate group on the adjacent sugar molecule. To initiate DNA replication, we need to break up these bonds. The enzyme helicase attaches to the origin of replication and separates the nucleotides. This separates the 2 strands of DNA. To stabilize the newly separated DNA, single-stranded binding proteins attach along each strand. As the helicase unwinds the DNA, torsional stress is induced along the DNA. To relieve the stress and prevent supercoiling, the gyrase enzyme binds ahead of the replication fork. The next step is elongation. A primase binds to the helicase and creates RNA primers, which allow for DNA polymerase III to add the corresponding nucleotides to the newly forming DNA strand. The nucleotides are added 5' to 3'. This creates a leading strand with continuous replication and a lagging strand that is created by discontinuous replication. The discontinuously created strands are known as Okazaki fragments. When the DNA polymerase III starts on a new Okazaki fragment, DNA polymerase I comes in and removes the RNA primer. This leaves a nick between fragments. To remove the nick, the enzyme ligase restores the sugar phosphate backbone and connects the Okazaki fragments. Once the ligase is removed, 2 strands of DNA are left. So, in conclusion, DNA replicates through initiation, elongation, and termination.